there are a broad of informations about each glial cells that you need to study further. This is just a really brief introduction. So in this lecture, our main objective is to learn more about the glial cells. But we also need to introduce briefly glial history and development and the CNS, then understand glial formation during development and the role of each individual CNS glia and microglia in health and in disease. So glial cells or neuroglia are non-neuronal cells in the nervous system that do not produce electrical impulse. So the term glia derives from the Greek glia, meaning glue. And because these cells were thought to be a kind of glue that fill up the space between neurons. But later, those cells were recognized for its importance within the nervous system. So the first event that eventually led to the discovery of glial cells was reported in 1543 by Vesalius, who firstly observed the difference between white and gray matter in the human brain. Later, glial cells and nerve cells morphology were described in 1824 by Dutrochet, who, however, identified them as components of a single anatomical unit. So almost 100 years later, in 1921, the first definition of the three main classes of glial cells in the central nervous system, which were astrocytes, microglia, and oligodendrocytes, were reported by Del Rio Ortega. And in the 1950s, the study of glial cells finally began and was detected resting potential and depolarization in glial cells, and additionally, new studies in new biochemical studies began to be further explored. So I couldn't ignore the important contributions of those two neuroscientists to the history of glia. So on the left, you have Monique Cajal. And although his contributions were mainly to the neuronal field, he also significantly contributes to the understanding of the nature of glial cells. And his drawings illustrated the brain cells and gave us an understanding of their morphology. So he was a Spanish neuroscientist and pathologist that focused on the histology of the central nervous system. And he and Camilo Gauge received the Nobel Prize in Physiology and Medicine in recognition of their work on the structure of the nervous system. And until date, Cajal is recognized as the father of modern neuroscience. Another important neuroscientist who also made great contributions to the field was Ben Barres. So he was a transgender and highly influential neurobiologist and advocate for women in science, and also a pioneer in understanding the functions of glia. Barres' major scientific contributions were related on how glial cells release factors that help to make synaptic connections between neurons. So glial cells are essential to maintain CNS and PNS homeostasis. So let's get into what are glial cells. So I thought I'd begin with some very broad statements that I think many of you already know. So the CNS is composed of a complex network of cells that control the receipt, processing, and response of information that is formed by the brain and the spinal cord. So the CNS is comprised by neurons and also glial cells. And neurons are cells with excitatory capacity, which electrochemical transmission is responsible for the conduction of information necessary for the development of the most basic functions in the animal kingdom, while glial cells are a diverse and heterogeneous classes of cells which support is essential for the maintenance of the nerve synapses as well as the physiology of the nervous system. So if glial cells are affected in somehow or they are not working properly, neurons probably will not survive. So glial cells are also known as neuroglia, and they were thought of as the supporting cells of the nervous system. However, recent evidence shows that they are not only supporting neurons, but they also they also protect and are actively engaged in brain homeostasis. So in the CNS, glial cells are a set of cells that display different functions. So they are derived from neuroectoderm cells, also called radioglial cells, which will give rise to most of the cells within the CNS, and also OPCs, which are the progenitors distributed throughout the brain that will give rise to oligodendrocytes and in some sort of astrocytes. And also by two classes of specialized cells, which are the microglia, which is comprised by oligodendrocytes, astrocytes, and ependymal cells, and microglia, which have a different embryonic origin from macroglial cells. So on the other hand, and talking very briefly, we have the PNS, which is comprised by neurons, and just one unique type of glial cells termed Schwann cells that have similar function to those displayed by oligodendrocytes. So we will learn more about each of those cells later on, but it's important to highlight that the current functions attributed to glial cells have been extended to include important roles in nervous systems that are beyond essential to neuronal functions. And although the absolute number of glial cells is still debatable in the field, it seems like it's very close to 1-to-1 ratio between neurons and glial cells. But where do glial cells come from? So before talking about the origin of glial cells, we need to remember some crucial steps inherent to embryogenesis. So as you may know, after fertilization, the single cell called zygote will undergo several mitoses for the development of the embryo, which will generate blastocysts. So the inner mass present within the blastocyst contain numerous pluripotent cells that will form the whole human body and the different organs and systems, including the nervous system. So the formation of the CNS and as well as all the tissues of our body is initiated from an embryonic stem cell, which would then give rise to the broad number of specialized cells present in the CNS nervous system. So the next step involved in embryonic development consists in the formation of 3 layers within the embryo. So the single layer blastula develops into a 3-layer termed ectoderm, mesoderm, and endoderm. So ectoderm is the outer layer in blue, mesoderm in red, and endoderm inner layer in yellow, each of which will give rise to different tissues in the human body. So if we zoom in this portion here and focus on the outlay, which is the ectoderm, we'll see that this structure will form the neural tube, the neural crest, and the epidermis. So which of them will give rise to a different tissue. So it's important to highlight here that the neural tube in early embryonic development is formed by neuroepithelial cells. So neuroepithelial cells form the wall of the closed neural tube, And the histologic terminology given by this is called neuroectoderm. So that's why we say that the glial cells within the CNS and neurons are formed by the neuroectoderm. So the neural tube cells will then become the neurons and the glial cells. And the neurocrest cells will give rise to the peripheral nervous system and therefore the Schwann cells. So on the other hand, another class of cells that has an important role in the CNS have a different embryonic origin. This class of cells is known as microglia, which is very distinct from other types of neuroglia. And microglia are derived from the yolk sac, which come from the hematopoietic stem cell progenitors and migrate to the brain as early as the 4th week of gestation in women. So the key point on this slide is that CNS glia is originated from the neuroectoderm, Schwann cells from the neurocrest, and microglia from the yolk sac. So we talked about the embryonic origin of each glial cells, and we learned that neuroepithelial cells from the neuroectoderm form the wall of the neurotube. So they are the kind of pluripotent cells, stem cells of the central nervous system in early embryogenesis. But it's important to remember that neuroepithelial cells have the ability to self-renew and line up the ventricles throughout at the stages of the neurotube closure. And they generate post-mitotic neurons, and also the intermediate progenitors known as radioglial cells or neurostem cells, which are asimilar but more fate-restricted stem cells. And radioglial cells will give rise to neurons, to oligodendrocyte precursor cells called OPCs, oligodendrocytes, astrocytes, and ependymal cells. So the radioglial cell is at the center of much of our understanding of a cortical organization during development. And the nomenclature radioglial cells arose because they resemble the morphology of astrocytes, but later they were described as neurostem cells. So in the adulthood, and the right side here represented, neurostem cells present in the subventricular zone will give rise to neurons, to OPCs, and astrocytes and ependymal cells. So OPCs are distributed throughout the brain and may also generate oligodendrocytes. And this is what we learn from here, that neurostem cells generate neurons, OPCs, oligodendrocytes, astrocytes, and ependymal cells in the CNS. So this slide is just a representation of the cortex, but obviously radioglial cells make up the whole brain. So this is a very busy slide, but the key message here is that something really fascinating in the embryonic development happens, which is neurostem cells produce neurons and glial cells in a strict spatio-temporal manner, where they generate all the neurons first, and then they switch to produce glial cells. So neurostem cells generate neurons first from embryonic age 12 to 18 in the mouse, and then they switch completely to only produce glial cells. So we have learned here so far that the CNS is comprised by neurons, stem cells, and progenitors that will give rise to most of the cells of the CNS except microglia. And those are derived from neuroepithelium or neuroectoderm. And you will learn that another distinct class of cells called microglia is originated from a different region termed yolk sac in the 4th week of gestation in humans. So on the other hand, the PNS is comprised by neurons, and one only glial cell known as Schwann cell, which is originated from the neural crest. So now we will talk about the functions of each glial cells, and we'll start with one of the most abundant glial cells termed astrocytes, which are mainly known for their function on blood-brain barrier maintenance and neuronal nutrition. So astrocytes were first thought to be the most abundant glial cells, but now accounts for the 20 to 40% of the glial population in the brain. Astrocytes are the stellate cells of the central nervous system that have numerous processes that make contacts with neurons and blood vessels. And astrocytes have characteristic star-like morphology, and that's why they are called astrocytes, because astro come from a star and cytes from cells. And they also regulate synapse formation, elimination, and function. They interact with neurons, regulating circuit functions, and have end feet that ensheath CNS vasculature, contributing to the metabolic support and homeostatic functions of neurons. They interact also with OPCs, microglia, and oligodendrocytes, relevance of which is still emerging. And those characteristics and feats that we just talked about are the terminal process that make astrocytes contact via tight junctions, blood vessels, neurons, and other cells. And regarding the blood vessel, this is important because completely seal off the blood supply within the brain. So astrocytes can take up nutrients from the blood vessels and transport to the neurons. So they are sort of messengers that help the communication between the blood supply and the neurons for exchange of molecules. So there are cells that present also immune properties and participate in the innate immune response within the CNS. And another crucial role of astrocytes is on regulating glutamate transport, and we will further explore this right now. Before exploring the role of astrocytes in the clearing of neurotransmissions, let's remind ourselves what is a synapses. Synapse is the site of transmission of electrical impulse between two neurons or between a neuron and a gland or a muscle cells. So it's just a region that allows a neuron to pass the information using an electrical or chemical signal to another neuron or another cell. So the synapse region is where the synapse happens, and it's termed synaptic cleft. So where the neurotransmissions, which are the chemical messengers, are released from the presynaptic neuron and binding to the receptors present in the postsynaptic neurons. This is a very simplistic way to show how a neurotransmission occurs. And this being said, it's well known that astrocytes can modulate brain activity, in particular through the uptake of neurotransmitters in excess. So for example, glutamate is the most abundant excitatory neurotransmitter that plays an important role in learning and memory. However, at high concentrations, glutamate causes an overexcitation of neurons, which eventually can lead to neuronal death. So here is the importance of astrocytes, because they project their end feeds in the synaptic cleft in order to control the excess of neurotransmission. And this functional integration between neurons and astrocytes is called tripartite synapse, because astrocytes actively participate in the synapse homeostasis. So it's important also to show that just about 20% of glutamate is taken up by postsynaptic glutamate receptors, while astrocytes uptake most of the remaining glutamates by their glutamate transporter, such as the excitatory amino acid transporters EAAT1 and 2, which are expressed on the surface of the astrocyte and the feet. So another thing is that glutamate could also be metabolized to glutamine by glutamine synthetase present in the astrocytes. And glutamine is then released in the extracellular space and can be taken up by neurons and used as a recycling mechanism to resynthesize glutamate. So on the other hand as well, glutamate can be within the astrocytes transformed into ATP. So another important function of astrocytes is involved in control of injury response. So during damage, astrocytes activation termed astrogliosis help them to migrate to the damaged site of the brain or the spinal cord, and there they will form glial scars around the damaged area, which main objective is to seal off the damaged area from the rest of the tissue, where there are lots of different toxic molecules. So in one hand, astrocytes are beneficial because they kind of seal off the damaged site. But on the other hand, they also secrete a number of molecules which are then causing a molecular program that prevents neuronal regeneration. So here's an example. So for example, after injury, damaged neurons could release molecules that will give signals to microglia. So microglia, activated microglia may release different signals such as inflammatory molecules that is gonna in turn activate astrocytes. And astrocytes, reactive astrocytes will start releasing also other molecules such as nitric oxide and other reactive oxygen species, which in turn leads to more neuronal death. Another important characteristic of astrocytes is that they are capable of display immune functions. So they are able of phagocytose cellular debris and actively respond against insult. And a marker of oligodendrocytes reactivity is when they increase the expression of glial fibrillary acid protein present in their cytoplasm. So let's now move on to another class of glial cell, which is particularly affecting multiple sclerosis, which is the oligodendrocytes. So we have learned that oligodendrocytes is part of the microglia and come from the neuroepithelium that cover the neuroectoderm. So we also learned that subventricular zone neurostem cells contribute to oligodendrocytes formation throughout life in normal brain and contribute to oligodendrogenesis, to the formation of new oligodendrocytes and remyelination in the demyelinated brain. So oligodendrocytes are the most abundant in the central nervous system. They comprise about 40 to 60% of the glial cells in the CNS and form the myelin sheath, which is an insulate layer made up of about 30% of proteins and 7% of lipids that wraps up axons. So the main purpose of myelin is to increase the speed at which electrical impulse propagates along the myelinate fiber, as you can see here on the bottom left image, in unmyelinated fibers, electrical impulse travel as continuous waves. Contrary, in myelinated fibers, they jump and therefore are much faster. So oligodendrocytes interacted closely with nerve cells and provide trophic support by the production of different neurotrophic factors such as GDNF, BDNF, and EGF1. So as an important feature is that a single oligodendrocyte can extend its process to over 50 axons, wrapping myelin sheath segments, which we call internodes, around each axon. So each oligodendrocyte forms one segment of myelin for several adjacent neurons. And diseases that affect oligodendrocytes are called demyelinating disease. So oligodendrocytes form myelin on axons to enable high-speed impulse transmission via saltatory conduction from one node of RNVA to another, increasing the velocity of axial potential. So nodes of RNVA are non-myelinated plates along the axons, enriched in ion channels that participates in the exchange of ions required to generate the axial potential. So another important thing is that astrocytes are in close contact with the axon at the nodes of RNVA regions, function of which is still unclear. So, and here is just to show up a transmission electron micrography of a mouse optic nerve, showing the multi-layers of compact myelin surround the axons, which is this structure inside that has lots of neurofilaments and microtubes on it. And nodes of RNVA are non-myelinated area along the axons. So we previously showed that OPCs generate oligodendrocytes throughout the adult brain and during development. So OPCs are the progenitors that also come from the neuroepithelium and have an important role in myelination and hyalination. So OPCs are the most proliferative cells of the CNS distributed throughout the adult brain that will give rise to oligodendrocytes and at some level astrocytes. And OPCs will receive different biochemical signals that will trigger their differentiation into mature oligodendrocytes. So a multipotent neuron cells here is expressing nestin requires different trophic factors to trigger the expression of transcriptor factors involved in OPCs specifications. Here we have a couple of them, such as OLIG2 and SOX8, 9, and 10. So once OPCs has acquired this specification and they start expressing A2B5, PDGF receptor alpha, and NG2. NG2 is a neuroglial antigen 2, and it's a reason why they're also called an NG2 cell. And differentiation along the oligodendroglial lineage to pre-oligodendrocytes or premature oligodendrocytes is potentiated by the addition of EGF1, CNTF, and T3 factors that are produced by different glial subtypes, and requires the transcription of different factors such as OLIG1, OLIG2, and SOX10 as well. And differentiation into mature oligodendrocytes is associated with the acquisition of myelin-related proteins such as myelin basic protein, MBP, and myelin-associated glycoproteins, MAG. Here's just a picture that was from my doctorate, from my thesis of NG2 positive OPCs, which show lots of branches throughout the, coming out from the soma. So in addition, several other biological activities also have been reported regarding OPCs heterogeneity and functions. So OPCs form synapses with excitatory and inhibitory neurons and have been shown as potential players in neuronal function, plasticity, and regeneration. They interact with many other cell types and extend their process, contact nodes of RNVA, receive synapses from axons, and regulate synaptic functions. So besides that, in the experimental autoimmune encephalomyelitis model, which is a model of multiple sclerosis, OPCs express genes involving the innate immune response, such as the histocompatibility complex class 1 and 2, called MHC1 and 2, which shows that they are capable to phagocytose myelin debris and may contribute to the pathogenesis of MS. So additionally, as they are highly proliferative and migrative, their nature have been explored as a tool to promote recovery in the myelinated disease, such as in MS. Because during injury, OPCs activate, proliferate, and migrate to the injury site, and generate new oligodendrocytes that are capable of remyelinating the CNS. And finally, OPCs also support the blood-brain barrier integrity via TGF-beta signaling. So currently, distinct phenotypes of OPCs and functional states have been reported, but more studies regarding the heterogeneity of these classes of cells is still needed to be addressed. So we talked about the different CNS glial cells and function, and we could not let behind the glial cells of the peripheral nervous system, although this was not part of the scope of the presentation, called Schwann cells. As we just talked about oligodendrocytes, I thought it would be the opportune moment to give it a very brief introduction on Schwann cells. So as we saw previously, Schwann cells are embryonically derived from the neurocrest, and they are the only glial cells within the PNS. So similarly to oligodendrocytes, Schwann cells myelinate axons, but within the peripheral nerves, and serve as the primary glial cells of the peripheral nervous system, insulating and providing nutrients to axons. So an important characteristic of Schwann cells is that each of them makes up a single myelin sheath segment on a peripheral axon, such that numerous Schwann cells are needed to myelinate the length of one axon, which is in contrast with oligodendrocytes, which form myelin sheaths for multiple axons. So in the same way, the compact myelin sheath insulates the axon segment, significantly increasing conduction velocity. So Schwann cells are critical in response to PNS axon damage and axon regeneration, and they myelinate PNS axon to provide metabolic support to neurons, but the only important caveat is that each Schwann cells really just makes up a single myelin sheath segment. So in fact, another interesting feature is that in the demyelination mouse model, Schwann cells can migrate into the CNS through blood vessels and myelinate CNS axons, functions of which needs to be better clarified. So let's get back to the CNS glia. We'll briefly talk about another kind of glia cells termed Ependymal cell, which is originated from the neuroepithelium coating the neuroectoderm during embryogenesis. So Ependymal cells form the epithelial line of the ventricles and the brain and the central canal of the spinal cord. And to talk about the glials, this is specifically a specific glial cell, we need to talk about the choroid plexus that are present in the lateral ventricles in the third and in the fourth ventricles. So each of the plexus is comprised of blood vessels with a single layer of ependymal cells joined by tight junctions, which forms the blood cerebrospinal fluid barrier. So the choroid plexus produce most of the cerebrospinal fluid, and the highly specialized ependymal cells of the choroid plexus produce the CSF by appropriate machineries for ionic transport and secretion of different molecules. So an important feature on ependymal cells is that they are postmitotic and derives from the neuroepithelium during embryogenesis. So ependymal cells are multiciliated cells that form the brain ventricular epithelium and provide an interface between the brain parenchyma and the ventricles in the CNS. And it's niche for neuron cells in the ventricular subventricular zone. But it's really hard to study ependymal cells because there is a lack of techniques that can distinguish ependymal cells from neighboring neuron cells. So it also has been suggested that ependymal cells filter brain molecules, insulate the brain from potentially harmful substance, and help the dispersion of neural messages in the CSF. So not too much have been reported besides their functions on CSF production and immunity. And currently here in Canada, Dr. Joanne Stratton is studying the functions of the ependymal cell to explore its role in multiple sclerosis and in neurodegeneration. And interestingly, ependymal cells, as they can act as a barrier, which probably means that they are the first responders to the environment that is in the cerebrospinal fluid. While the cells are understood, as Dr. Wee Young explained in the first Alberta MS network lecture that we had, MS lesions are usually located close to the ventricles. And so perhaps ependymal cells are involved in the onset or pathogenesis of MS. So there are many things to uncover regarding ependymal cells function. And last but not least, I will switch my gears and talk about microglial cells, which are originated from the yolk sac. So as microglia come from a different origin, they are quite different in function. So microglia are the resident immune cells of the brain, and in addition to immune surveillance roles, microglia is also constantly monitoring brain activity by extending and retracting their highly ramified process. And they also regulate numerous developmental and functional processes, including synaptic pruning, clearing apoptotic cells, and interacting with multiple CNS types in health and in disease. Additionally, microglia also secrete a brood of molecules involved in CNS homeostasis. So the so-called resting or quiescent microglia constantly and actively scan their environment for exogenous or endogenous signals that may threat the CNS homeostasis. An important aspect of microglia is that they continue to self-renew differently from macrophages, which are the same origin of microglia. However, they don't have the ability, but that they are continually regenerate from the bone marrow. So we already showed the embryonic origin of microglia. I just want to highlight here that the studies show that microglia are distinct from bone marrow-derived monocytes. So they are derived from primitive macrophages that emanate from the yolk sac during development prior to embryonic day 8.5. and enter the brain via the circulatory system. So these microglia progenitors surround the neuroepithelium of the developing brain around the embryonic age 9.5, and one day later, they enter the neuroepithelium and begin to colonize the CNS parenchyma. So microglia exhibit heterogeneity in the transcriptome, protein expression, proliferation, and function, which might be involved in their pro-regenerating properties. So these are just images from drawn by Pio de Rio Ortega, which show microglial cells evolution during their phagocytic activity. So they present different morphologies that reflects their activation status. And homeostatic microglia are highly ramified and continually scan the parenchyma. So upon brain injury that induces cell death, damaged tissue and surrounding astrocytes will release extracellular adenosine triphosphate, which is a molecule that provides energy to drive many processes in living cells, but can also act as a messenger. But when it's in high concentrations in the extracellular environment, triggers activation of a specific purinergic receptors in microglia, for example, and which in turn is gonna induce the extension of process of microglia towards the cell that is injured, and which later will result in the cleanup of cellular debris. So microglia can also, at this stage, release different pro-inflammatory cytokines, engage in antigen presentations, and trigger the arrival of also peripheral immune cells. But the cellular debris removal is an important step for controlling the extent of the injury, because it's well known that cellular debris are important inhibitors of differentiation in the CNS. And besides, during the demyelination, microglia supports the recruitment of OPCs and their subsequent differentiation into mature myelinating oligodendrocytes through clearance of myelin debris, secretion of growth factors and cytokines, and modulation of the extracellular matrix. So here's just a video showing the highly phagocytic nature of microglia cell, which plays an important role in CNS homeostasis. So in addition to everything we learned today, it's important to highlight the involvement of glial cells on inflammation. So inflammation is a physiological and important mechanism to contain insults. However, when the inflammatory response is exacerbated, it can be irreversible. So in the brain parenchyma, neuroinflammation occurs as a vicious cycle with the participation of glial cells that make up the CNS. So as an example here, in response to first neuroinflammatory stimulus such as LPS, so the recognition of the antigen is initiated through the toll-like receptor 4, highly present in the membrane of astrocytes and microglia, which in turn will activate several transduction pathways, and at the end cause the activation of NF-kappa B, which will result in increased secretion or increased transcription of genes encoding pro-inflammatory cytokines, such as from the IL-1 family, and which in turn can lead to neuronal death. So as neurons and oligodendrocytes have cytokine receptors, especially the ones from the IL and TNF family, these cells are very susceptible to damage. So here is just to show that the exacerbation of inflammation has inhibitory signals that it will cause, for example, other cells to differentiate. For example, in MS, OPCs fail to differentiate because there are lots of signals given by different immune cells in the CNS. So demyelination, which is the loss of oligodendrocytes, will lead to exposure of the axon and lately degeneration, which characterize the late stages of MS. So as a take-home message here, we learned so far that within the CNS, glial cells are formed by neurostem cells and OPCs, which are the progenitors of the CNS, and by oligodendrocytes, astrocytes, and ependymal cells, which are the microglia and come from the neuroepithelium. And microglia which originate from the yolk sac. So the glial cells of the PNS are the Schwann cells, which are capable of forming only one myelin segment per axon. So neuron cells will give rise to neurons, astrocytes, and oligodendrocytes, and has different functions that I didn't mention here because it's a lot of information, but they also participate of immune control of the CNS and involved in the migration and differentiation of the CNS glia. OPCs will give rise to oligodendrocytes and astrocytes and participate of the immune response of the CNS, releasing trophical factors that can control other cells' activities, such as microglia as well, and support blood-brain barrier integrity. Oligodendrocytes are the myelin-forming cells of the CNS that wrap up axons, allowing action potential conductions and provide metabolic support to actions. Microglia are the professional antigen presenter cells of the CNS that regulate synaptic circuit remodeling and phagocytosis, and their main function in neurodegenerative disease is the removal of abnormal extracellular proteins or debris that may inhibit differentiation. And they play an important role upon controlling OPC's fate through secreting a brood of molecules involved in OPC's differentiation. And when there is an exacerbation of inflammatory response, microglia may play deleterious effects on the CNS. So Ependymal cells lines up the choroid plexus, forming an interface between the brain parenchyma and the ventricles, produce CSF, and have an important role in filtering brain molecules. And astrocytes, which tightly regulates blood-brain barrier formation and maturation, participate in the regulation of synapses, control the excess of neurotransmitters, and similarly to microglia, during injury may play an important role in the exacerbation of the inflammatory response. So that's it. I hope you have learned a little about glial cells, and many thanks for your time.
Thank you, Monique. Really well done.
Thank you
so much, Monique. Are you able to see the questions in the chat box?
Let me see 1 second. Okay, so the first question from Manor, I'm sorry if I'm not pronouncing it right. Why Schwann cells myelinate axon in 1:1 ratio? Is it the cell intrinsic behavior or cell extrinsic? Well, it's like there are lots of different information regarding Schwann cells' ability to myelinate axons. So basically, there are different clues given to Schwann cells, such as the production of different growth factors that is going to give signal to axons in to the peripheral axons to myelinate specifically one segment. We don't know, and I'm not like acquainted with Schwann cells, honestly. So maybe like it's the axon, the peripheral axon per se, that gave the signal to Schwann cells to myelinate just one myelin segment. So yeah, so when we have in the peripheral nervous system, the Schwann cells will myelinate to myelinate a whole axon. We need lots of different Schwann cells, and this is I don't think it's completely elucidated. So yeah, if there is someone that works with Schwann cells on here, please give your input. I think it's going to be okay. So next question. Each myelination cell, some are non-myelinate, line up axon along the axon and envelop it. One Schwann cell wraps one segment of an axon. Its nucleus is on the outside. Yes. do you think the microenvironment in the CNS and PNS can also contribute to the different myelination program by the cells? I think the microenvironment in CNS and PNS can also, yes, is what I just said. I think the microenvironment, the difference between the PNS and CNS may give different signals that is gonna make Schwann cells have the ability to myelinate just one axon, while the oligodendrocytes can myelinate multiple axons. So the environment of CNS and PNS is just starting to be interrogated, yes. So Anastasia gave us I think it's a nice overview on the difference between the environment on CNS and PNS, which is probably why Schwann cells are able to myelinate. But like the publication I showed, let me just go back here. Yeah, this is very interesting because so this publication, for example, they show that Schwann cells can migrate into the myelinated brain and myelinate axons, but they are still not able to myelinate lots of axons at the same time. So they myelinate generally just one segment of axon. So it doesn't seem as just related to the microenvironment, since they don't acquire new properties once they are in the CNS. It seems like there is something else related to the Schwann cell per se. Okay. Next question was, what do you know about differentiate and then redifferentiation? For instance, an astrocyte becoming a progenitor and then maturating to an OPC, is that possible? Yeah, there are some publications talking about the progenitor state of astrocytes. We're not totally confident about the role of astrocytes as a progenitor, but I honestly think that everything is possible, since like we can reprogram a cell entirely and make them become a progenitor or vice versa. So I truly think that is possible to make a cell become a progenitor. So you mentioned that microglia may have detrimental effects on neurons by promoting inflammation, but is there any evidence that a loss or lack of microglial function is detrimental in neural health? Yes, surely. Microglia participate in the homeostasis of the central nervous system. That's a very good question. So including, as I said, in the synaptic pruning during development. So there is a researcher called Beth Stevens at the University of Harvard, and she works closely with the understanding of synaptic pruning by microglia. So microglia play an extremely important role for CNS maintenance, including during inflammation. This is a important process to contain the damage. So inflammation per se is not bad. The problem is when inflammation is exacerbated for signals that we still don't know. Like sometimes for reasons that are still unclear, we don't have the completely control of the micro environment, and therefore other cells of the CNS boost more inflammation. For example, neurons per se can release different cytokines that can activate astrocytes, microglia, oligodendrocytes, precursor cells. So it's microglia, if you, for example, if you have mouse lacking microglia, it's not going to survive. So microglia really plays an important function in brain homeostasis. You're welcome. So anything else?
More questions?
If anyone else want to give an input, like feel free. Dr. Wee?
I've already asked my question. I was going to type to thank you for a very thorough presentation. I thought that you covered a broad area really, really well and brought it all down to concepts that and information that we can all understand. I thought it was very well done.
Oh, thank you so much. I'm happy to hear.
Okay. Thank you, Monique. And
Thank you, guys.
we'll see everyone next Thursday.
Okay, bye.
Okay. Thank you again.
Thank you, bye.
Bye.
Stay well, everybody.